Today we'll be exploring Lyme disease, one of the most common vector-borne diseases in the United States. Specifically, we'll focus on the innate and adaptive immune responses to Borrelia burgdorferi. Even though it's not very common here in Las Vegas, it's a great example of how the immune system responds to infection and why those responses aren't always enough. Lyme disease is caused by the bacterium Borrelia burgdorferi and is transmitted through infected tick bites. While we often refer to it as a single bacterium, Lyme disease is actually caused by a group of closely related Borrelia genospecies. A genospecies refers to a genetically distinct group within the same type of bacteria. Four of these genospecies are responsible for most human infections, and within them, there are about 6 serotypes, which are variations based on differences in surface antigens. These antigen differences are important because they can affect how the immune system recognizes the bacteria and may contribute to immune evasion. Infection progresses in 3 stages. In the localized stage, the bacteria enters the skin and begins to spread locally, causing symptoms like fever, fatigue, and a bullseye rash. Early disseminated stage, the bacteria is spread through the bloodstream. And if untreated, this infection progresses to the late stage where the bacteria will persist in the tissues and can lead to arthritis, cognitive issues, and heart inflammation. The immune response begins with the innate immune system, which provides the first line of defense and helps initiate adaptive immunity. Dendritic cells and macrophages recognize the bacteria through pattern recognition receptors and respond rapidly. Once activated, these cells release inflammatory cytokines such as TNF alpha and interleukin 6. These cytokines promote inflammation and recruit additional immune cells to the site of infection. Dendritic cells also present antigens, which is what helps activate the adaptive immune response. Before looking at the data, it's important to understand that dendritic cells respond to infection by becoming activated and releasing cytokines. TNF alpha is measured because it is a key cytokine that promotes inflammation, recruits other immune cells, and helps initiate adaptive immune responses. In the context of Lyme disease, this early activation is important for controlling bacterial spread and priming T cells. In this experiment, human dendritic cells were cultured and exposed to different conditions. Starting with the control, this is the unstimulated group, so the cells were not exposed to any stimulus, which gives us the baseline of TNF alpha. Next is the positive control, which was treated with LTA, a known activator of dendritic cells. Then we have Borrelia burgdorferi, strain 31. These cells were exposed to bacteria at a specific MOI, or multiplicity of infection, which is the ratio of bacteria to cells. Based on this setup, we expect to see increased TNF alpha in the bacteria are activating the dendritic cells. Looking at the results, TNF alpha levels are the lowest in the control group, which is expected since there is no stimulation. In the positive control, TNF alpha levels are much higher, confirming that dendritic cells are capable of responding. In the Borrelia burgdorferi group, TNF alpha levels are also significantly increased compared to the control and also similarly to the positive control. These values are also highly significant. From the immune system perspective, this shows that dendritic cells recognize the bacteria and respond by producing inflammatory cytokines. This is important because it represents one of the early steps in the immune response and helps initiate activation of adaptive immunity. However, in the context of disease, the strong inflammatory response does not guarantee bacterial clearance and may contribute to inflammation and symptoms while the bacteria persist. From this data, we could see that Borrelia strongly activates dendritic cells. After activation, dendritic cells process and present bacterial antigens on HLA-DR molecules to T helper cells. At the same time, cytokines released by dendritic cells and other innate immune cells help promote activation of lymphocytes. Other innate immune cells involved during this stage are macrophages, which help by phagocytosing bacteria and releasing inflammatory cytokines. Neutrophils contribute to earlier bacterial clearance. Natural killer cells produce interferon gamma, which helps activate macrophages and dendritic cells. And mast cells also play a role by releasing histamines and chemokines that reduce additional immune cells to the infection site. As this process continues, T helper cells become activated and begin coordinating the adaptive immune response. The adaptive immune system provides a more specific response to infection. Antibody-mediated immunity is a key part of that response. After activation, B cells differentiate into plasma cells that produce antibodies targeting Borrelia. IgM is produced in early infection and helps control bacteria in the bloodstream, but it is less specific. IgG is produced later, but is more specific. and helps limit the spread of infection into tissues. These antibodies work by binding to the bacteria, which can neutralize them or mark them for destruction by other immune cells through a process called opsonization. However, even with antibody production, the bacteria are not always fully eliminated, which contributes to persistence of infection.
Before looking at the data, it's important to understand that not all B cells are the same. B cells are a critical part of the adaptive immune system and are responsible for producing antibodies that help neutralize pathogens and limit bacterial spread. B1 cells develop early and create a more innate like response, meaning they respond quickly but are less specific. B2 cells are the conventional adaptive B cells and are responsible for producing more targeted pathogen specific antibodies. B1 cells mainly produce IgM early in infection, while B2 cells produce IgM first and then IgG later as the response becomes more specific. The distinction is important because in the data we'll look at next, the researchers are comparing which of these B cell types is actually responsible for producing Borrelia specific antibodies. In this study, mice were infected with Borrelia, and the number of Borrelia specific IgM producing B cells in the lymph nodes was measured. The results showed that B2 cells produced significantly higher levels of IgM compared to B1 cells. This demonstrates that adaptive B cells are actively responding to the infection, confirming activation of the adaptive immune response. Functionally, IgM antibodies appear early in infection and help control bacteria in the bloodstream. IgG antibodies appear later, are more specific, and help limit the spread of infection throughout the body. Overall, these results show that the B2 cell response is critical for controlling early infection, but additional immune mechanisms are required to fully eliminate bacteria from tissues. Although the immune system mounts both innate and adaptive responses, there are important limitations. From the innate data, we saw strong activation of dendritic cells and high TNF alpha production, but this alone does not guarantee that the bacteria are clear. From the adaptive data, IgM antibodies are produced early, but they are not sufficient for clearing bacteria from tissues. IgG antibodies develop later and improve control of the infection, but even those more specific antibodies do not fully eliminate the bacteria. One important immune evasion mechanism is antigen variation. Borrelia can change the proteins on its surface, which are the main targets that antibodies recognize. These surface proteins or antigens can vary between different serotypes or even change over time during infection. When the immune system produces antibodies against one version of the antigen, the bacteria can switch to a different version. Because of this, existing antibodies may no longer recognize the bacteria efficiently. This allows the bacteria to avoid immune detection and continue surviving in the body, even when an immune response is already activated. So, because the immune system isn't always able to completely eliminate Borrelia, the infection can spread to different parts of the body, including the joints. This is where Lyme arthritis comes in. Lyme arthritis is a late stage complication of Lyme disease that occurs when the bacteria infects joint tissues and triggers inflammation. The most noticeable symptom is swelling in one or a few joints, most commonly in the knees. The joint may also feel warm, painful, or stiff, and the swelling can sometimes come and go or even move between joints. This condition usually develops weeks or months after the initial infection. The good news is that Lyme arthritis is treatable. Most patients improve within a 4-week course of oral antibiotics, especially when it's caught early. However, if it's not treated properly, it can lead to permanent joint damage. In some cases, symptoms may persist even after treatment, but this is usually due to the immune system rather than an ongoing infection. To better understand the immune response in Lyme arthritis, researchers used an ELISA test to measure antipeptidoglycan IgG levels in synovial fluid samples. These samples came from 82 patients and were blinded, meaning the researchers did not know which samples were from Lyme arthritis patients and which were from other types of arthritis, like rheumatoid arthritis. Peptidoglycan is a major component of bacterial cell walls, so antibodies against it help indicate an immune response to bacterial infection. The results showed that IgG levels were significantly higher in patients with Lyme arthritis compared to the control group. Additionally, in Lyme arthritis patients, IgG levels were higher in the synovial fluid than in the blood. This suggests that the immune response is not just happening systematically, but it's actually concentrated in the joints, which is the main site of inflammation. These findings indicate that patients with Lyme arthritis produce significant antibodies targeting bacterial components, and that this response is localized to the affected joints. After a tick bite, Borrelia enters the skin, which marks the beginning of infection. The innate immune system responds first. Dendritic cells and macrophages recognize the bacteria through pattern recognition receptors and release cytokines, which promote inflammation and recruit other immune cells to the site of infection. Activated dendritic cells then process and present bacterial antigens to T helper cells, which help initiate the adaptive immune response. T helper cells activate B cells, leading to an antibody production. IgM is produced early as a rapid but less specific response, followed by IgG, which is more targeted and helps control the infection. However, despite these immune responses, the bacteria can evade the immune system by changing its surface proteins and hiding in tissues. As a result, the infection may persist, leading to an ongoing inflammation, tissue damage, and later stage complications. Clinically, this is important because Lyme disease doesn't just stay localized. It can spread throughout the body and affect multiple systems, including the joints, heart, and nervous system. The immune response plays a dual role. It helps control the infection, but it also contributes to inflammation, which can cause many of the symptoms we see in patients. Even though the immune system produces antibodies, including IgM and IgG, these do not always lead to complete clearance of Borrelia. As a result, the bacteria can persist in tissues, leading to an ongoing immune activation and chronic inflammation. This could explain long term complications such as Lyme arthritis and other persistent symptoms. The variation in surface antigens across different Borrelia strains can also impact how efficiently the immune system responds in patients. This may contribute to differences in disease severity and challenges in fully clearing the infection. Understanding these immune mechanisms is important because it can help improve how we diagnose, treat, and manage Lyme disease.